Welcome to Module 1 of Introductory Biochemistry. This course provides a detailed foundation in the chemical principles underlying living systems. As we begin this journey, we will focus on the history and scope of biochemistry, the structure of cells and membranes, and the chemistry of essential biomolecules. Biochemistry is often referred to as the chemistry of life. It forms the bridge between biology and chemistry, and it plays a fundamental role in understanding how our bodies function at the molecular level. Let's begin with a look at the historical background and definition of biochemistry. Lecture 1: A brief history and definition of biochemistry. Biochemistry as a formal field of study began in the 19th century. However, the exploration of the chemical basis of life has roots that go back centuries. Early studies on fermentation and digestion hinted at the presence of biological catalysts, now known as enzymes. In the early 20th century, scientists began to understand the roles of proteins, carbohydrates, and lipids. The discovery of DNA's structure in the 1950s marked a significant leap, linking genetic information to molecular biology. Biochemistry can be defined as the study of the chemical substances and vital processes occurring in living organisms. It examines the molecular mechanisms that govern life, from energy production and enzyme activity to genetic coding and cellular signaling. Lecture 2: Importance of biochemistry in medicine and science. Biochemistry is a core discipline in medicine, pharmacy, agriculture, and biotechnology. It helps explain how diseases occur, how drugs act, and how nutrients are processed in the body. In medicine, biochemistry provides insight into metabolic diseases such as diabetes, genetic disorders and mutations, hormonal imbalances, cancer development, drug action and detoxification. In other fields, it aids in food production, crop enhancement, environmental protection, and the development of industrial enzymes and vaccines. Lecture 3: The living cell, organization and molecular architecture. The cell is the basic structural and functional unit of all living organisms. Biochemistry focuses on what happens inside cells, the molecules, reactions, and structures that make life possible. There are two major cell types. Prokaryotic cells: these cells lack a nucleus. Examples include bacteria and archaea. Eukaryotic cells: these have a nucleus and membrane-bound organelles, including mitochondria, endoplasmic reticulum, Golgi apparatus, and lysosomes. All plants, animals, fungi, and protists are made up of eukaryotic cells. Each cell contains DNA, RNA, proteins, and various other biomolecules arranged in highly organized ways to ensure survival and reproduction. Lecture 4: Biological membranes, structure and organization. Biological membranes are essential components of cells. They separate internal components from the external environment and control the passage of substances in and out of the cell. Membranes are made up primarily of phospholipids. These form a bilayer that provides the structural framework. Proteins, embedded in the membrane to facilitate transport and communication. Carbohydrates, attached to proteins, and lipids, playing roles in cell recognition. The fluid mosaic model describes the membrane as a dynamic structure where molecules move laterally, allowing flexibility and function. Membranes also create compartments within eukaryotic cells, allowing for specialized environments and reactions in organelles such as mitochondria and the nucleus. Lecture 5: Biomolecules and the origin of life. The origin of life is closely tied to the development of organic molecules. It is believed that life began when simple molecules combined to form more complex ones under early Earth conditions. Four major classes of biomolecules emerged: carbohydrates, lipids, proteins, nucleic acids. These molecules serve as the building blocks of cells. Over time, the interactions between these molecules led to the formation of early life forms capable of replication and metabolism. Understanding these biomolecules is central to studying biochemistry. Lecture 6: Carbohydrates, structure, classification, and functions. Carbohydrates are organic compounds made of carbon, hydrogen, and oxygen. They are a primary source of energy for living organisms and also play structural roles. Classification of carbohydrates. Monosaccharides, simple sugars like glucose and fructose. Disaccharides, formed by two monosaccharides, such as sucrose and lactose. Oligosaccharides, short chains of sugar units. Polysaccharides, long chains like starch, glycogen, and cellulose. Functions include providing energy, serving as structural components in plants and animals, acting in cell recognition and signaling. Carbohydrates also react chemically in processes like oxidation and condensation. These reactions are vital for metabolism and energy production. Lecture 7: Lipids, essential fatty acids and fat-soluble vitamins. Lipids are a diverse group of hydrophobic molecules. They include fats, oils, steroids, and phospholipids. Key among them are essential fatty acids, which the body cannot synthesize and must be obtained from the diet. Two major essential fatty acids are linoleic acid, alpha-linolenic acid. They play important roles in inflammation, brain function, and cell signaling. Eicosanoids are signaling molecules derived from essential fatty acids. They regulate functions such as blood clotting, immunity, and smooth muscle contraction. Fat-soluble vitamins include vitamins A, D, E, and K. Their structures allow them to dissolve in fats and oils, and they are stored in the body's fatty tissues and liver. Each has distinct functions. Vitamin A supports vision and immune function. Vitamin D regulates calcium metabolism. Vitamin E acts as an antioxidant. Vitamin K is essential for blood clotting. Lecture 8: Proteins, peptide bonds and structure. Proteins are complex molecules made of amino acids linked by peptide bonds. Their structure determines their function. There are 4 levels of protein structure. Primary structure, the sequence of amino acids. Secondary structure, local folding into alpha helices and beta sheets. Tertiary structure, the overall 3D shape of a protein. Quaternary structure, the assembly of multiple polypeptide chains. Proteins perform many vital roles. Catalyzing biochemical reactions as enzymes. Transporting substances like oxygen. Supporting cell structure. Enabling muscle contraction. Regulating biological processes through hormones. Any alteration in protein structure can lead to dysfunction and disease. Lecture 9: Nucleic acids, DNA and RNA. Nucleic acids store and transfer genetic information. There are 2 main types. Deoxyribonucleic acid, the DNA stores genetic information in the nucleus. Ribonucleic acid, RNA helps in decoding DNA into proteins. DNA is a double-stranded molecule with a unique sequence of nucleotides. Each nucleotide contains a sugar, a phosphate group, and a nitrogenous base. RNA exists in several forms, such as messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, RNA. Each plays a role in protein synthesis. Understanding DNA and RNA is essential to grasping genetics, inheritance, and cellular regulation. Summary of Module 1. In this first module, we have covered the origins and importance of biochemistry, the structure and types of cells, the organization of biological membranes, the role of biomolecules in life, the classification and function of carbohydrates, lipids, proteins, and nucleic acids. These foundational concepts are critical for understanding how biochemical processes maintain life and health. In Module 2, we will focus on enzymes, vitamins, and their coenzymatic functions, along with the clinical implications of deficiencies and toxicities. Welcome to Module 2 of Introductory Biochemistry. In this module, we will explore enzymes, the biological catalyst essential to life, and vitamins, the organic molecules crucial to metabolic regulation and well-being. We'll also address how imbalances in these compounds can lead to diseases or health complications. Let's begin with enzymes. Lecture 1: Understanding enzymes, nature, structure, and function. Enzymes are specialized proteins that accelerate chemical reactions in living organisms. Without enzymes, life would not be possible, as most biochemical reactions would proceed too slowly to sustain life. Enzymes are highly specific, each one typically catalyzes a single chemical reaction or a group of closely related reactions. They work by lowering the activation energy required for a reaction to occur. Enzymes act on substrates, the molecules they convert into products. This transformation occurs at the active site of the enzyme, which fits the substrate like a lock and key. Some common examples of enzymes include: amylase, breaks down starch into sugars. Lipase, digests fats. Protease, breaks down proteins. Lecture 2: Zymogens, enzyme specificity, and the active site. Some enzymes are synthesized as inactive precursors called zymogens or proenzymes. These must be activated by specific biochemical changes before they can function. For example, pepsinogen is activated to pepsin in the stomach to begin protein digestion. Enzyme specificity refers to the enzyme's ability to select a specific substrate and catalyze its transformation. Specificity is determined by the structure of the enzyme's active site, which complements the substrate's shape and charge. There are 4 main types of enzyme specificity. Absolute specificity acts on only one substrate. Group specificity acts on molecules with similar functional groups. Linkage specificity acts on specific types of chemical bonds. Stereochemical specificity acts on specific enantiomers or isomers. Lecture 3: Enzyme inhibition and activation. Enzyme activity can be regulated through inhibition or activation. Inhibitors are substances that reduce enzyme activity. They are categorized into: competitive inhibitors, bind to the active site, competing with the substrate. Non-competitive inhibitors, bind to another part of the enzyme, changing its shape. Uncompetitive inhibitors, bind only to the enzyme substrate complex. Enzymes may also be activated by cofactors, which are non-protein substances that enhance enzyme function. Cofactors include metal ions such as magnesium or zinc. Organic molecules called coenzymes, which are often derived from vitamins. Enzyme regulation allows cells to conserve energy and respond to changes in the environment or metabolism. Lecture 4: Factors affecting enzyme activity. Enzyme-catalyzed reactions are influenced by several factors. Temperature: each enzyme has an optimum temperature at which it functions best. Too high or too low a temperature can denature the enzyme. pH: enzymes have an optimal pH range. For example, pepsin works best in acidic conditions, while trypsin functions in a basic environment. Substrate concentration: increasing substrate concentration increases reaction rate up to a point. Enzyme concentration: more enzyme typically results in more product, assuming substrates are available. Presence of inhibitors or activators: these can slow down or speed up enzyme activity. Lecture 5: Allosteric enzymes and isoenzymes. Some enzymes have allosteric sites, regulatory sites other than the active site. Binding of a molecule to an allosteric site can increase or decrease enzyme activity. These enzymes are essential in feedback regulation of metabolic pathways. Isoenzymes, or isozymes, are different forms of an enzyme that catalyze the same reaction but differ in structure and may be present in different tissues. For example, lactate dehydrogenase has several isoenzymes specific to the liver, heart, or muscles. Understanding isozymes helps in diagnosing tissue-specific damage. For instance, elevated cardiac isoenzymes may indicate a heart attack. Lecture 6: Introduction to vitamins. Vitamins are organic compounds required in small amounts to support normal metabolism and physiological function. They are not synthesized in sufficient amounts by the body and must be obtained from the diet. There are two major groups: fat-soluble vitamins A, D, E, and K. Water-soluble vitamins B-complex and C. Each vitamin plays a specific role, and deficiency or excess can lead to health issues. Lecture 7: Vitamin Deficiencies and Related Conditions. Let's examine some important vitamin-related health problems. Vitamin A deficiency can lead to night blindness and dry skin. Vitamin D deficiency causes rickets in children and osteomalacia in adults. Vitamin E deficiency may result in nerve and muscle damage. Vitamin K deficiency impairs blood clotting. Among water-soluble vitamins, vitamin B1 thiamine deficiency leads to beriberi. Vitamin B12 deficiency can cause pernicious anemia. Vitamin C deficiency results in scurvy, marked by bleeding gums and poor wound healing. Understanding these conditions helps in diagnosis and dietary management. Lecture 8: Vitaminosis, Hypovitaminosis, and Antivitamins. Three terms are key in understanding vitamin balance. Vitaminosis, general term for disorders caused by abnormal vitamin levels, either too much or too little. Hypovitaminosis refers specifically to vitamin deficiency, often due to poor diet, malabsorption, or chronic disease. Antivitamins, substances that interfere with the absorption or action of vitamins. For example, avidin in raw egg whites binds biotin, preventing its absorption. In some cases, medications or environmental factors can act as antivitamins. Lecture 9: Vitamins and their coenzyme functions. Many vitamins serve as precursors to coenzymes, which are vital for enzymatic reactions. Here are a few examples. Vitamin B1, thiamine, becomes thiamine pyrophosphate, used in carbohydrate metabolism. Vitamin B2, riboflavin, forms part of FAD and FMN, important in oxidation reduction reactions. Vitamin B3, niacin, contributes to NAD and NADP, essential in energy metabolism. Vitamin B6, pyridoxine, is needed for amino acid metabolism. Folic acid and vitamin B12 are crucial for DNA synthesis and red blood cell production. Without these coenzymes, many biochemical pathways would be disrupted. Lecture 10: Biomedical Importance of Vitamins. Vitamins play essential roles in metabolism, acting as coenzymes in reactions involving energy production. Immunity, vitamins A, C, and E support immune system function. Tissue repair and growth, vitamin C aids collagen synthesis, while vitamin D supports bone health. Neurological function, B-complex vitamins are vital for nerve function. Reproduction and development, vitamins A and D are involved in fetal growth and development. In clinical practice, vitamin levels are routinely monitored in patients with malnutrition, chronic diseases, and certain medication regimens. Module 2 Summary. In this module, we explored the structure and function of enzymes, zymogens, enzyme activation and inhibition, factors affecting enzyme activity, the importance of allosteric enzymes and isoenzymes, classification and functions of vitamins, deficiency diseases, hypovitaminosis, and the role of antivitamins. Vitamins as coenzymes and their biomedical relevance. Conclusion of Introductory Biochemistry. Congratulations on completing the course Introductory Biochemistry. You have now developed a foundational understanding of the molecular basis of life, the structure and role of biomolecules, the importance of enzymes in catalysis, the critical roles of vitamins in health and disease. This course sets the stage for deeper studies in medical biochemistry, molecular biology, nutrition, and related disciplines. Your understanding of these core concepts will be instrumental in your academic and professional development in the life sciences.